Thanks, Tony. Good afternoon, everyone. For those of you that don't know me, my name is Jeff Klein, and it's my honor and privilege to be the Executive Director of the ALS Association Greater Philadelphia Chapter. Thanks for joining us today for this question and answer session about ALS research, which is a follow-up to the recent Help and Hope Symposium. If you weren't able to join us for this symposium, it was fully recorded and can be accessed at any time on Facebook and YouTube. So we'll get right to it. It's now my pleasure to introduce Ellen Phillips, as Tony mentioned, who is currently our board chair emeritus, but has had many titles throughout her decades of being a champion for the ALS cause. And Ellen will be introducing our special guests. So thanks again for coming, Ellen.
Thank you, Jeff. Pleasure to be here. And I'm glad that we have these sessions. I think that they're critical. I was on the presentations last week, and the speakers are fantastic and very easy to understand, which has always been a bugaboo for me. Sometimes when listening to some scientists, I feel like I need to go to school, medical school, just to be able to understand, but I don't find that that was the problem at all. So here we go. I will introduce all three speakers, and Tony is the moderator. He will direct questions to our specific researchers or to all of them. So Zachary Simmons is well known to all of us in our region. He is the professor and vice chair for research at the Department of Neurology and the director of the ALS Center at Penn State Health. He also is the editor of Muscle and Nerve. His interests are primarily focused on ALS. He's published extensively about the quality of life in ALS and was the co-developer of the ALS-specific quality of life instrument. Related to this, he has published on ethics and care and end-of-life issues, telemedicine, including telemedicine, both synchronous video conferencing and asynchronous, I may not be pronouncing that correctly. Now I'll have to go back to English class. Remote monitoring initiatives are being pursued in collaboration with colleagues in engineering and speech and communications. These include initiatives in home pulmonary function testing, monitoring of gait and falls, and monitoring of speech and swallowing. Cognitive and behavioral dysfunction in ALS are also other active areas of Dr. Simmons' research, including the use of an instrument to measure apathy and the study of a mindfulness program to improve quality of life and to decrease anxiety and depression. Dr. Simmons is also actively involved in clinical trials and has served as a site principal investigator for many studies during the past 2 decades. The multidisciplinary ALS clinic at Penn State Health was founded by Dr. Simmons with tremendous support from our nurse, Susan Walsh. He continues to direct this and it is an ALS Association certified treatment center of excellence where more than 200 patients with ALS and other motor neuron disorders receive regular care from a plethora of highly trained group of physicians, nurses, and healthcare professionals. Patients with related disorders such as primary lateral sclerosis, progressive muscular atrophy, progressive bulbar palsy, and hereditary spastic paraplegia are also seen regularly. Dr. Simmons continues to provide ongoing care for patients with a variety of other neuromuscular disorders and to perform electrodiagnostic studies, which are nerve conduction studies and electromyograph or EMG. Oh, we're still going on. There's going to be no time for the other speakers, sorry.
Feel free to stop.
I'm going to stop, but not before I say that you're board certified in neurology, American Board of Psychiatry and Neurology, 1989 board certified in electrodiagnostic medicine, American Board of Electrodiagnostic Medicine, and you also hold a joint appointment in the Department of Humanities at Penn State College of Medicine. And you are not 150 years old after all those credentials. So thank you, Zach. Appreciate you being here.
Thank you, Ellen.
Next up on our roster is an extraordinary woman, Lucie Bruin, who has a PhD and an MBA. She is the Therapeutic Area Lead for Novartis Institute for Biomedical Research, Biomarker Development Department at Novartis. Lucie Bruin has worked in neurodegenerative field for the past 28 years with a specific interest in developing therapies for these devastating disorders. As therapeutic head for ALS at Novartis, she oversees therapeutic strategies and biomarker development for antibody, small molecule, and gene therapy assets. Prior to joining Avexis, Gene Therapies, which is now Novartis, as she was chief scientist of the ALS Association for 20 plus years. As chief scientist at ALSA, she established the first translational research program for ALS. That's something to take note of. Translational research advances therapies for ALS through partnerships between academia, industry, and government, and solicits donor contributions for strategic programs. She established initiatives in drug development, clinical trials, biomarkers, assistive technology, precision medicine, large-scale sequencing, and analytics. These initiatives directly led to the development of antisense technologies for neurodegeneration and have contributed towards the tools required for industry to tackle the challenges of drug development for ALS. Dr. Bruin received her BSc in pharmacology at Rhodes University in South Africa, her MSc in neuroscience at King's College in London, and her PhD in biochemistry, focusing on tau biology and Alzheimer's disease at King's College, London. More recently, she received her MBA, traveling on planes across the world, at Imperial College, London, with a focus on industry, academic partnerships, and the role of intellectual property. Thank you, Dr. Bruin. And certainly not least in the three people that we're talking about, last but not least, is Dr. Sabrina Paganoni, MD, PhD, co-director, Mass General Hospital Neurological Clinical Research Institute. Dr. Paganoni is an assistant professor at Harvard Medical School and works as a physician scientist at the Healy Center for ALS at Mass General Hospital and at Spaulding Rehabilitation Hospital. Her research focus on developing new treatments for ALS that's her focus is on developing new treatments for ALS. And she designed and is currently leading several ALS clinical trials that include novel endpoints and biomarkers and innovative trial designs. She is currently the co-principal investigator of the Healy ALS platform trial, the first platform trial for ALS in the world. And I think you've all been hearing a lot about the Healy platform trial. She is also working with people with PLS, primary lateral sclerosis, a rare form of motor neuron disease, and hereditary spastic paraplegia. She co-chairs the PLS Task Force and the PLS Registry of the Northeastern ALS Consortium. We know it as NEALS. She is working with other Harvard researchers on developing new assistive technology technology devices that can improve functions and quality of life for people with ALS and other forms of motor neuron disease. Dr. Paganoni has published over 75 peer-reviewed papers. She's an editorial board member for Muscle and Nerve and the American Journal of PM&R, where she is also associate editor for the section Evidence-Based Physiatry Cochrane Corner. Her research has been funded by the NIH, foundations and industry. She received several awards for her work, including the NIH Rehabilitation Medicine Scientist Training Program Award in 2012, the AAN/ALS Association 3-Year Career Development Award in ALS in 2017, and the American Association of Neuromuscular and Electrodiagnostic Medicine Scientific Impact Award 2019. Dr. Paganoni holds 3 board certifications: physical medicine and rehabilitation, neuromuscular medicine, and electrodiagnostic medicine. She is a member of the Association of Academic Physiatrists, the American Academy of Neurology, and American Association of Neuromuscular and Electrodiagnostic Medicine. Those are our 3 presenters. Reading all that they do, I'm not sure how they had the time to be with us today, but thank you very much for being here. And now I will turn this over to Tony, who is our moderator, and will ask questions that have been submitted.
Thank you, Ellen, and again, thank you everyone for participating. We obviously have a very great group of presenters. So I want to ask one of the questions that have been sent around. This is for all 3 of the researchers or anyone that wants to speak from the 3 presenters. First, have researchers learned things from the COVID-19 pandemic that may be useful for ALS research as well?
I can certainly start to comment, but we'll then let Sabrina and Zach. I mean, from our perspective now working at Novartis, we've learned that really we have to spend more and invest more in digital technologies, and that actually being able to serve people with ALS remotely and being able to make it easier for participation in trials is really pivotal. So that is one learning. As much as I'm sure it's great to be able to go into clinic, there are lots of bonuses to being able to do things at home. So that was a key lesson for us internally as a team, and I'm sure that Zach and Sabrina can expand on that.
Yeah, thank you, Lucie. So we've been, I would agree 100% with what Dr. Bruin said, that we've also seen this as a time when we've been, in some respects, forced, but in a good way, to look to other ways of communicating with and helping to monitor our patients without being able to have the physical contact that we sometimes do. I don't think by any means that video communication and remote monitoring is a substitute for all in-person visits, but it certainly can augment them. And I think one of our main take-homes was during the height of the COVID pandemic, we found that we could still communicate with and have visits with 90% of our patients, either using telephone or video or some combination of the two. And now that the pandemic is at a more stable state, we still find that about 30% of our patients come to see us remotely, simply because they can't make the trip physically, or they could potentially, but they don't have the resources to get into clinic, or some combination of the two. So the digital technology has really been extraordinarily helpful from a patient care point of view, and as Dr. Bruin pointed out, certainly from a research point of view as well.
Yeah, I'll just add to that. I mean, I agree with what both of you have said, so I'm not going to repeat that. One message that I think is very important for the audience and people living with ALS is that ALS research has not stopped during the pandemic. In fact, I would say that we have seen a lot of progress, lots of projects that continue despite the pandemic, and they evolved and adapted using digital tools, video monitoring, etc. So we've seen a lot of work that has been going on thanks to really the dedication of all participants, all sites. Everyone has been involved with research. many of the lessons learned will stay with us, and I hope that will also provide some improvements, such as the long-term incorporation of telemedicine and digital monitoring, which can have some positive aspects. And we're still seeing some disruption from the pandemic. Interestingly enough, at this point, I think a lot of the disruption is also related to supply chain difficulties. So I just want the community to know that there is a lot, there's really a team of people, lots of teams around the country, around the world, trying to continue this research. Research has been going on, will continue to get better. We learned from the pandemic, but there's still some disruption because of some, unexpected circumstances like supply chain issues.
Thank you for that. Speaking of trials that are continuing, all of you probably can discuss this. How do studies like the Healy platform trials open up research to more people with ALS? And will this be a benefit to other diseases as well, potentially?
Yeah, sure. Yeah. So if I understand the question is, sort of like the fact that now we are enrolling more participants. We're sort of, if I understand the question correctly, like how we are actually including more people and broadening access. So I think that that's really something that we've been working on with the patient community for a long time, and we're so excited to open up to a broader population. And I think this will have a lot of repercussions, I think, on many other trials, not just for ALS, but also for other disease models. In fact, we have active discussions. We have several that already happened and some that are upcoming with other disease consortia, other neuromuscular diseases or neurological diseases. They want to learn about how to engage with the patient community, specifically how to be responsive to the need for greater access. And again, we continue this conversation with different consortia. So I think both short-term and long-term, it will have an impact on other disease areas as well.
Again, please, if you're not one of the presenters, please mute your microphone. Dr. Simmons, do you have anything to add? And I know you've been working on this as well.
Yeah, I was going to just say that all of Sabrina's points are important ones. The other thing I was going to add is just from a very sort of an operational point of view, the way that the Healy study has been designed, and Dr. Paganoni should take a great deal of credit for how much work she's done on this study. The way that the study is designed, it's done in such a way with the input from patients that it has lowered the bar for entry into the study. For example, the requirements for breathing capacity are lower for this study than they are for many other studies, allowing more people to be able to qualify for the study. It has a 3-to-1 drug to placebo ratio, meaning that 3 times as many people who are entered into the study receive the drug as the placebo. So that's something that encourages some of our people who participate to be more likely to participate because of that. It also has a long open label extension, so that after people are done in the double-blind portion of the study, they receive open label drug if they choose to do so for a long period of time afterward. It's been opened up at multiple sites across the country, many more sites than we usually have for most clinical trials, improving access. And there's been a reduction, in the number of in-person visits that are needed as some of the data is collected remotely. So I think in addition to the overall broad sort of view of how do you open up trials to more people, this is a really good practical aspect of a study that has done just that and has just made the study more, not only more accessible, but more desirable to a large number of people.
No, thank you for what you said. I as you were talking, I also wanted to mention something else that I think is important to discuss on a broader level. We know that historically, only about 10% of people with ALS have been participating in research, and that number has been relatively stable over time. So I think, again, it's been roughly that figure. And what we have learned from other fields of medicine is that there are other disease areas, other, specific diseases where the participation is much higher. So really what we are trying to do with the Healy platform trial, but also with other trials and other projects that many of the other speakers are part of, the idea here is to really change the framework where we want to allow people with ALS who want to participate in research to do so. So we're basically creating sort of a high volume, a level participation, as opposed to, just one or two people can participate. And that's a very dramatic change of kind of a shift in practice, because basically this means that everyone who wants can contribute in one way or another. And so this creates also the need to think about, and we're working on that. Certainly that's something that we pay a lot of attention to, like in terms of how do we best allocate resources so that we really bring this up to the next level, where the goal is for the majority of people with ALS who want to participate to be able to participate, whether it's in a formal trial, whether it's an expanded access process or other types of research, biomarkers, etc. So we want to create a landscape where this is sort of part of the culture. And so again, it's going to take some time to get there, but I think we really want to continue to work towards allocating resources and supporting sites and all the different clinics that many of you work with, to be able to offer that type of high volume participation.
Thank you. Lucie, you can explain on that as well, but I have two questions here I'd like to get to next. First from Lee Jablow. And he asks, I am concerned that many of the investigational therapies in development are being studied in advanced ALS. It's sort of like showing up to a large house fire with a small fire extinguisher. I wonder if therapies that would prove ineffective in late ALS may prove highly effective in very early or pre-symptomatic ALS, specifically as this relates to familial ALS and work on biomarkers in asymptomatic patients. What are your thoughts on studying these unique pre-symptomatic populations have a better sense of impact on prevention or delay of ALS onset? And if effective, how might this line of thinking apply to sporadic cases? And this is open to any one of our presenters.
I'm happy to tackle that. We obviously all spend a lot of time thinking about that. I think there are multiple parts to that question. Absolutely, absolutely correct to look at a pre-symptomatic population. In fact, Biogen's doing exactly that. There's a trial running now for the SOD in a pre-symptomatic. And the exciting reason why that is even possible is that we have indicators in the population that some of the key biomarkers are already elevated prior to any clinical signs. So neurofilaments, which is really probably for ALS, one of the most, at this point, most validated soluble biomarkers, is something you can measure in the blood and the spinal fluid, is shown to be elevated both in those families that carry the C9 mutation years before onset of symptoms, and an SOD population as well, several years before onset. So definitely in those populations where we know the genetics, we definitely will be, and we are doing preclinical studies in patients that have not shown any symptoms yet. And so you're absolutely right. The earlier you can go with the treatment, the more likely we'll have a success. That said, of course, the majority of ALS, I think we sort of move away from the terminology sporadic because we know that genetics is involved to some extent in other cases of ALS where they're not linked directly to a single gene mutation. But in cases where the genetics is not well known, there's no family history, it's very difficult to go in early because the likelihood is that person will only present at the clinic when there are signs and the disease is already ongoing. But we also, a lot of our time, think about maybe there are different pathways involved at different stages of disease. And I know we have at Novartis one particular asset that we are testing, which is looking at inflammation. And maybe inflammation is a key pathway much later in the disease or at least after onset. So there might be reasons why some treatments do actually work at a later stage. We know also on the platform, and Sabrina can make further comments, there are anti-inflammatory type compounds. So I think absolutely the earlier we can come in with a treatment, that's why we invest in biomarkers very heavily. But where this is not possible, there still is a potential for therapies to have an effect. So others, Zach and Sabrina, please comment.
Yeah, so thank you, Lucie. So I think there's a bit of a natural tension here between, on the one hand, as Sabrina mentioned, Many people who would like to participate in ALS research either have not been able to or simply have not wanted to participate in ALS research. So for those individuals, what we're trying to do is to break down barriers to participation. And even though it does seem as if the people who are being entered into ALS are already advanced in their disease, there is a real need for individuals who have clinically apparent disease to have treatments offered to them, and as Lucy was just saying, and in fact, the reason that most individuals don't qualify for trials, or one of the main reasons, is that their disease is too advanced, and so there's really been an effort on the part of clinical researchers to get people in early, but that doesn't answer your main concern, which is, should we, from a research point of view and from a knowledge point of view, should we be trying to get them in even earlier in order to try to prevent or to get in at the very beginning and learn how we can do that? But it's a challenge because ALS is a rare disease. So it's not as if it occurs in so many people that we can do routine blood screening of the entire population and identify individuals who may be at risk to develop ALS, because it occurs in such a small percentage of the population overall. Certainly the work that's been done in SOD1 and C9orf ALS does give us some very good guidelines of how we can screen people who are pre-symptomatic in advance. The bigger question of, can you screen people where there's no family history of ALS and identify those in advance and start treatment in advance? That's a tougher question in a rare disease, I think.
Yeah, I don't have much to add because both of you explained this so well, but perhaps just, again, to think about in analogy with other disease areas, if you think about cancer, for example, we know that there needs to be, or there's been a need for combination treatments for a long time until targeted treatments then became available. And then as targeted treatments became available for one form of cancer, then it was clear that it could be translated to other forms of cancer that share similar molecular mechanisms. All of this to say that I wouldn't, I think we really need to continue to do research on all aspects of the disease, familial, sporadic, early, late, because again, there's multiple mechanisms at play, and to get the highest chances of success, we really need to tackle all of them. And it's not going to be a one size fits all, but it's probably going to be a combination of treatments for that particular person. And I think that, we're not in this situation yet, but very soon we will be in a situation similar to what, some cancer patients experience, or people with multiple sclerosis, to mention another neurological disease, where the physician will have to work with the patient to discuss which combination and at what time is most applicable to that particular person at any given time. So it's not going to be kind of a one drug that solves it all, but it's going to be probably, I would imagine that maybe 5 years from now, we'll have a number of treatments that can be combined in different ways, and it will be more personalized than it is today.
Thank you. This next question comes from Pennsylvania State Representative Kyle Mullins. And Kyle, I will break the rules and I'll let you, do you want to ask this live, Kyle, or would you like me to just read it? Because you may want to provide some background on why you're asking it.
Sure. First and foremost, I want to thank you, the experts, whose contributions to fighting this awful disease are I take quite personally, and owe you personal thanks, although we've never met. That's what's heaven sent about people like you. You dedicate your life and your time and your education to trying to save people who hundreds and thousands you'll never meet. My father was diagnosed with ALS in January of 2020, and by the skin of his teeth, his breathing capacity, he was accepted into the Healy platform trial out of Penn in Philadelphia. He's actually going for his 3-month checkup tomorrow, and he's holding his own. But you all know how tough this is. But I've long been interested from my position as a public official in rare diseases, especially in providing, making sure that government is doing all it can to provide the necessary resources for not only research, but also patient care. But long story short, I know we have some, of course, some expertise in the Healy trial here, but question regarding AMLX. What is the current status and timeline for AMLX? And, as a follow-up, and, as I understand it, AMLX and eventually, hopefully, one of the Healy drugs can be taken in combination, but what's the timeline that we're likely to understand or glean some information from the Healy trial in which regimen or regimens appear effective or promising?
Yeah, I'm happy to start taking the AMLX question, as I've been, involved with the trial as the principal investigator. So, obviously now the company is doing a lot of work to accelerate, all the next steps. As you may know from public announcements and recent press releases, the company has announced the intention to submit to the FDA for a new drug application, also known as an NDA. So that's what we know, that the process has started, which is great news. What we do not know is the exact date of submission or how long the review process will take at the FDA level. So historically, again, with no relationship to this particular trial, but historically it takes a few months for the FDA to accept a submission for review and then file it and then sort of review it until ultimately, hopefully the drug will be approved and go be available on the market. So that's sort of the general sort of status and timeline for AMLX. And I would say that, it's really been amazing to see this program, come together. I know Dr. Bruin knows this well because she was involved as the chief scientist officer from the ALS Association around 2015, 2016. We wrote a grant to the ALS Association, and that's what started this program. In other words, this program would have never taken off had it not been because of the work that the ALS Association and especially Dr. Bruin has done in terms of providing the initial seed funding that then led to lots more work. So this is to say that this has been a community collaboration from day one, really even at the time of design of the trial, and now continues to be a great collaboration. And I know AMLX Pharmaceuticals is doing everything they can to bring this to completion. So I think that that's the status and timeline. Does that answer your question?
Yes, very much so. Thank you, Doctor.
Thank you. Yeah, in terms of your other question, and obviously I'm happy for the other speakers to weigh in as well. First of all, I'm really glad that your father can be part of the platform trial. We know that the platform trial is really, again, another community project where the participants are not just participants, they're actual valued research partners. They are making this, they're making the history of ALS happen in collaboration with us. And so the platform trial is a mechanism for testing as many drugs as possible in the shortest possible period of time. And also, I would say another scientific goal is to also learn more about ALS in the process. So it's not just about testing drugs, it's about testing drugs, testing them as fast as possible, and contributing to the field as we do that. So I think it's a win-win, on every front. Now, the question is that, unfortunately, as in any clinical trial, you really don't know which regimen or regimens is more promising. The promise comes from the science, and the science and the preclinical data are what makes us interested in a product. But then it's only when the trial reads out at the end that we know which one is going to be positive or not. And the idea is to continue to add more regimens. So again, to test as many as possible. Now, as we recently discussed, we have weekly webinars, by the way. So we continue to update the community on the progress every week. And we launched the trial right after the pandemic started in mid 2020. And we are going to have the results of the first 4 drugs in 2022. So in 2 years, basically, we have results for 4 drugs, not 1. Now, if you think about the history of ALS trials, if you just look back, this is a complete shift in speed. It's a complete acceleration, because to have results for 4 drugs, not for 1, over this time frame means that we are really making everything so much faster. And so while I cannot answer your question in terms of which regimen will be positive, I can tell you that we are certainly going to learn if it's positive or not. We designed it in collaboration with the FDA to test specific features of each drug, which is very important. Not all trials are created equal. Actually, you have to design them to answer specific scientific questions. And this was really designed in collaboration with the FDA and industry partners and the patient community to answer very key questions. And so this is to say that we're going to learn a lot. And I really hope that, at least one of the regimens will be positive, if not more. And in any case, more regimens will be added. But what we do know is that we have, again, started this process of testing many drugs quickly and enabling broader participation. So at least that's a, important next step.
Great, thank you so much. I have a question from Wendy Bear. In last week's presentation, environmental factors were mentioned as being contributory to the development of ALS. In my community, there have been 5 ALS diagnoses within a 2-mile radius within the last 8 years. For a rare disease, that seems significant. Can any of you comment on location as a factor, as well as other factors like excessive exercise, as contributing factors to the development of this disease? That's open to everybody.
So, the geography has been a tough factor to implicate clearly in the epidemiology of ALS, because there have been a number of factors in different studies that come out showing that those factors are associated with a somewhat higher frequency of ALS, but none of them are really sort of hit it out of the park, home run types of associations. These are associations in which the chance of getting ALS is increased somewhat in individuals who have those sorts of exposures. One of the more classic ones is long-term exposure, for example, to certain pesticides. Particularly as a child, there's been an association in some studies, such as in Italian soccer players, with head injuries. And then the association with athletic ability or sports ability, the papers are really all over the place. There are a number of them that clearly show some sort of positive association between what I'll call cardiovascular health and a tendency to be somewhat more likely to get ALS. But again, none of the associations are of the magnitude that you would see between, for example, cigarette smoking and lung cancer, or some other obvious associations. The cluster of ALS that you describe there have been a number of clusters, and I'm not an epidemiologist, but certainly there have been a number of clusters of ALS described in the past at various locations. The challenge is that random clusters happen, and for example, there are also areas that one could identify that within a 2-mile radius have been ALS free, for 100 years. And one would then ask the question, well, isn't that unusual? What is it that's protective about that area? So while clusters are helpful, unless we can identify common factors that are present in all those clusters, it becomes really, really hard to make a determination, particularly when the associations are present but not super robust. And that's why I think that although environmental factors may act as triggers in some sort of way in people who are genetically predisposed to develop ALS, I think that it's what we're most likely seeing is that it's not the main factor, but that some factors may be triggers, but that a genetic or other predisposition most likely has to be there.
I would just be happy to add, I know that in my presentation, I put in an image that is work of Dr. Amaral Chalabi, who describes it, I think, really well as a stepwise disease. So in other words, the risk if you have the family gene is a big chunk of the risk to get, and dictates when you might get onset of the disease, and environmental factors might play into that. So just echoing what Zach said, single environmental factors are very unlikely to cause the disease. And in fact, it's hard to ascertain exactly how they impact it, but it could be just triggers that on top of other genetic susceptibility, you then are more likely to get ALS. And so that was referenced in the slide and the work of Dr. Amaral Chalabi.
Thank you so much.
Thank you. This next question comes from Christine Moretti, and she asks, if a drug was introduced into the Healy trial that was specific to UMN treatments, would PLS patients be included? And would length of disease determine if a patient would be accepted? Thank you.
Yeah, this is a multi-part question, I think, and also would love to hear from the other speakers as well. So as of right now, I'm not aware of any investigational product that's specifically targeted to the upper motor neurons. I know there's been some research coming up from colleagues at Northwestern and other areas where there's a few agents in development that might be targeted to upper motor neurons. And I think, they could be tested in a standalone trial or in the Healy trial. Basically, if we were to develop a trial, if there was the availability of a treatment to be tested in a trial for upper motor neuron predominant forms of the disease, then obviously one would want to enrich or enroll those people specifically. So people that have signs and symptoms of upper motor neuron disease, which are specific and can be evaluated as part of the clinical exam to some extent. Now, there is a specific presentation of motor neuron disease where the upper motor neuron predominance is particularly pronounced. Essentially there is the presentation is limited to upper motor neuron problems, and that's called PLS, which you're asking about specifically. So that PLS, when you have that specific presentation, it tends to progress at a much slower rate than people with classic ALS or more mixed presentations. And because of that, I think, many of us, I know many of us on the line are working on developing trial designs and tools to really assess PLS, because it might require very specific assessment tools, because pure PLS tends to behave in a little bit of a different way in terms of outcomes, meaning it progresses very slowly, and so it has a very specific presentation. So I think, to answer your question, I think, again, I don't think of any trial, I don't know of any trial-ready agent that could be tested to only target upper motor neurons. If one was available, that would be great, and we would definitely be able to test it in a variety of trials, whether it's the Healy trial or other trials. And specifically, if there is an even more specific or targeted treatment for PLS, then that would require specific trial designs, which we're working on. I don't know if others have other comments.
I would just echo Sabrina. I'm not aware of any ready compounds for just upper motor neurons.
And I would also agree with everything that was said. The one other thing I would add is when one looks at the therapeutic landscape right now, the challenge is twofold. It's how good are the drugs that we have available, and how good are the tools that we have to measure the effects of those drugs. If we had an experimental compound that we were relatively certain would improve strength, then I think there would be no reason not to use everybody with any form, to permit everybody with any form of motor neuron disease to participate in those trials, because we can easily measure improvements in how people are doing. On the other hand, the drugs that we currently have are designed with the expectation primarily that they'll slow progression. If a disease is progressing only at a very, very slow rate, like primary lateral sclerosis, then as Sabrina said, the part of the challenge is the tools that you have available to measure that. And although we are looking at things that might be more sensitive than, for example, the ALS functional rating scale, or we're looking at other biomarkers like neurofilaments, as Lucie mentioned, then potentially those could be used to measure it. But right now, we're really left without good tools to measure extremely slow progression in a disease where the best we can do is to slow progression by a bit.
Thank you for those responses. And for anyone that has a question that you want to ask, we have a few minutes left, so you can put it in the chat. I'm going to ask one more, and then if there's another question that comes in, we will ask that as well. So this is probably for all of you, but maybe Dr. Simmons specifically. What benefits from telemedicine have you seen that have impressed you the most? Is this the future of both care and research for ALS?
Sure. Well, thank you for asking that. So I'll be brief, and I think it does play a role in both care and research. I would say it's a part of the future, not the entire future. And I'm not saying that you're implying that in any way, whoever asked the question, but I think it's a part. And from a care point of view, there are now, we're looking at telemedicine as a way of simple, straightforward video communication between people with ALS and their healthcare providers. That's a great step because it allows us to see people toward the end of their disease when they otherwise couldn't make the trip in. And it also allows us to regularly see people who simply don't have the resources to come to ALS clinic, even if they don't live very far away. So it also permits one-on-one visits with healthcare providers through video visits, and that can facilitate better care as well. From a research perspective, I think there are really two ways that it can help. One is the way that both Lucie and Sabrina have commented on, which is that we can reduce the number of in-person visits potentially by collecting some data remotely, and that allows more people to be in the study with less burden to them. There are also a number of apps that are being designed in order to allow patients with ALS to communicate more freely with their healthcare providers and to exchange more information. Apps where they record their functional rating scale score, they record their weight, where they record their voice and send that in. And then I think lastly, from a research point of view, this allows us, and this has clinical implications, this allows us to do some monitoring so we can be more proactive. So if we could put a wrist sensor or an ankle sensor on people and detect that their walking is changing, and then have our physical therapists see them early because they're showing signs that they're getting at higher risk for falling, that would be very important. Or if we can record their voice remotely, hear some changes that put them at high risk for aspiration and bring them in earlier to see our speech therapist, that would be important as well. So yeah, I think there are a lot of implications. I think we've just started to scratch the surface of this.
I would just add to that, Zach, not only from a care perspective, but of course, we are, as you indicated, really looking for other biomarkers, whether they're the neurofilament-like soluble markers, but actually, and I'll highlight a company, Oral Analytics, who I believe are also working with the Healy platform. Being able to analyze the voice might also then, if you have an intervention that's successful, can then measure the changes in the voice analytics. And interestingly, the company has also shared some early data that you can see changes in cognition on voice analytics, and that helps potentially as an outcome measure for trials like the C9. So as much as it also will indicate when care is needed and to prevent things in care, it can also guide us in drug development.
Thank you. I don't know if you have anything else to add, Dr. Paganoni, but the other questions I have were already answered or combined with some other questions. So if any of you have any closing statements to make about the hope you see in the progress of research in the last year, I think that would be a great way to close this discussion.
Is there an opportunity to ask a question?
Yes, there is. I was asking people to type in, but there's still a chance to ask one more question.
Oh, can I ask it now?
Yes.
Okay, well, you know, with the Human Genome Project that was sort of completed in 2000, the CRISPR-Cas process has come in, and it allows you to go in and if you identify a defective gene that is a cause of something, you can replace it, as has been done already in certain cases. I think sickle cell, for example, I believe, and replace it with a normal gene. And so I was wondering, it seems that if there's been some progress in identifying some of those defective genes that may be greatly contributing to ALS, you probably haven't found all of them yet, but you have found maybe some of them. And has there been any attempt to use the CRISPR-Cas process to go in and actually replace those genes with a normal gene to see what effects you might have?
I'm happy to take the comment. So absolutely, this technology is very much being used. And I would sort of backtrack a little bit and say, obviously, before that technology, antisense was a real opportunity. And in fact, through the association, the funding of the association, it was the first time that that technology of antisense was used in a neurological disorder, and teams at Merit and Sabrina and Tim Miller and others really brought that forward into the clinic. Following on from that, there's of course gene therapy efforts. It was what attracted me into working with the Vexus gene therapies. But there are approaches to doing that for those genes like SOD1 and C9. So absolutely, for those where those things can be done, either it's adding in a gene, it's changing a gene, it's changing the splicing of a gene. And Biogen has moved forward with their technologies into some genes that are maybe risk factors like ataxin. So absolutely, this is very, very doable. I think the challenge will be that in the cases where there's not a single gene mutation, but there are multiple interacting factors, this could be more tricky. But I think it's a really good question, and it's technology that will be applied and is in process. I suppose it leads me, before others want to make quick comments, to the comment that I thought would be an important one for the Greater Philadelphia, for people with the disease, living with the disease, just to recognize the contribution that you all have made and make as people living with it, or people that have given funds to the research programs. Just being in a slightly different position now at Novartis, where I do also work with ALS and work with a portfolio, we benefit internally enormously from the data that's been generated and from those studies. And so I just highlight Project Mine, Onset ALS, GTEC, very large initiatives collecting materials from people living with the disease, from their genetics, whether they're sporadic, whether they have family history, or the clinical data. So we can do just what you are indicating, find more genes for the disease, start to understand subpopulations of people that will respond to certain therapies. So for me, it's very gratifying to see how at a very local level, each one of you have impacted the field so enormously. And now it's just a real pleasure to be able to say to colleagues within the company, well, actually the ALS field have experts from the scientists and the colleagues we see on the phone today, to clinical centers globally, to materials we can access. So we really are in a unique position. I say the we, as I often have used in the past, because it is a collective, it is a global effort, and it is thanks to you at the very local level that have made such an impact. So keep participating, whether it's in biomarkers, whether it's in collecting information from a clinical level, because it all together has made an enormous impact on the disease. And for that, I'm very, very grateful. So that was a segue from your question into my comment as to the impact and the real future for ALS as a disease.
Oh, one final comment. May I ask it? Oh, well, sorry.
Yes, if it's a
absolutely.
Very quick
I'll answer for Tony.
Yes, if it's a very quick comment.
Oh, well, the reason why I asked it in part was that my daughter, who is at the University of Delaware, has been doing some of that in gene therapy, in molecular, that sort of thing. And she's done some work on that. So that's the reason it motivated my question, that if they can start doing it in certain cases, like in Huntington's and sickle cell, that there's some hope for also approaching that with, ALS, which is apparently much more complicated, of course.
Yeah, perhaps I'll leverage that last comment for a few final thoughts. Absolutely, it is a collective, as Dr. Bruin said, and we're really learning from people who are doing research in other areas. So I think that what your daughter does and what researchers in other fields do, they do all contribute. I do think, I really think it's important for people to understand how many people are now working on ALS and learning from other fields, as well as, the fact that we are collaborating and bringing other people from other fields into ALS. And that's because of the tremendous advocacy and interest, I think, really with the Ice Bucket Challenge and all the work that happened after that. A lot of that work was also done by the ALS Association or projects funded by the ALS Association. That really changed things in terms of the number of people interested in the disease from a scientific perspective. And that's important because it's only where there's funding and there's interest that there can be progress. And so the fact that we're all continuing to promote the disease, raise awareness, have campaigns, fundraising efforts, legislative efforts, research projects that continue to basically amplify the message. And as we bring more people into ALS, the answer will come from different places, even places we don't know about yet. And new technologies will continue to be incorporated. So thank you for that comment.
I'll just make one brief closing statement as well. All of us who work with individuals who have ALS are well aware that that time is of the essence, and I think other than individuals and their families themselves, I think I speak for most physicians and scientists in the ALS community when I say that other than you, nobody wants a cure for ALS more than we do. I think the encouraging thing currently is that there are more drugs and more studies going on for ALS now than at any time that I can ever remember. And I'm quite certain that the number of clinical trials going on now and the number of drugs being tested exceeds that at any comparable period in time. So that's all been a very, very positive step. I realize it's not a cure. But I agree very much with what Sabrina said earlier, which is that I think that it won't be so much as eureka, we've got a cure, as well we have another drug and then another drug and then yet another one, which when combined have a really meaningful effect on the overall course of ALS. And I see us making progress toward in that direction at this point. And from my perspective, it's very encouraging. And I echo everybody's thanks to the to people with ALS and to their caregivers and the rest of the ALS community, because our work wouldn't be possible without your support.
Thank you to everyone participating. Thank you to our researchers for the wonderful answers. For everyone listening, you can find this video and other videos on our YouTube page at ALS Philadelphia and on other social media. And please learn more about our chapter and our services at www.alsphiladelphia.org. Thank you very much and have a good day.
Thank you, everyone.
Thank you. Very, very fine discussion. Thank you very much. Very good.
Thank you.